everybody. We're going to get started now. I'm David Strauss. I'm one of the conference co-chairs, and it's a pleasure to invite you today welcome you here today to panel 4, which is called Considerations of Race and Ethnicity in Research and Research Protections. Some people are still wandering in, but we're going to go ahead and get started. We're really pleased. I'll tell you a little bit about the sort of the genesis of the panel, just in brief, and do a brief introduction of our two panelists, and then we'll get going. You know, as the conference planning committee reflected on the conference theme, which is the 40-year history of this organization of Primer, and the essentially 40-year history of the field since the National Research Act, the notion of race in relation to research and research ethics emerged as one that was especially important, we think, over the past 40 years, and one that has evolved over time and warranted some reflection in a panel at the conference. We also noted that it is a notion or an issue that remains singularly complex, and hope today to talk or touch on at least a couple of aspects of that complexity. Certainly when we think about race and research, at the extreme end, we can all catalog what I think we'd all agree is the fairly grotesque history of exploitation of individuals from various racial and ethnic groups in the name of science, or research conducted to demonstrate racial inferiority, or harmful research conducted without regard to the special vulnerabilities or sensitivities or preferences of specific ethnic or racial communities. Today, less shocking but still problematic, much of the work that we do in academic medicine, both research and research oversight, occurs in academic and research institutions and in IRBs that are perhaps inadequately diverse, that do not reflect the populations we study or serve. And we need to ask the question, how confident can we be that we can ask the right questions? We record the demographics of our study samples in research in search of representativeness or generalizability. But how effective are we at ensuring that we actually recruit and study samples that permit us to appreciate racial differences in disease and treatment response? How good are we at that? There are many challenges, therefore, before us, and today we really want to focus on one particular axis of debate. And that's that, on the one hand, there may be enormous value in examining research questions along racial lines, that identifying evidence of race-specific treatment responses or failure to respond is or may be enormously important. And on the other hand, on the other end of that debate, is the notion that race is a social construct, and that research that focuses on race in many ways may be problematic. So let me move ahead now and introduce our two speakers who I am delighted to introduce and have with us here. First is Dr. Ann Taylor. Ann is a professor of medicine and cardiology and senior vice president for faculty affairs and career development for Columbia University Medical Center, and she's vice dean for academic affairs at Columbia's College of Physicians and Surgeons. Ann received her medical degree from the University of Chicago School of Medicine and completed her internship residency and clinical cardiology fellowship there, and did research training at Johns Hopkins and the University of Iowa. Dr. Taylor's interests include cardiovascular disease in African Americans and women, the transfer of principles of cardiovascular disease prevention from academia to the community, and professional development for faculty. Dr. Taylor was the chair of a steering committee for the African American Heart Failure Trial, a national multi-center trial of nitric oxide enhancing therapy in congestive heart failure. I'm going to go ahead and also introduce Charmaine Royal. Professor Royal who's an associate professor of African and African American Studies and director of the Center on Genomics, Race, Identity, and Difference at Duke University. Dr. Royal received a master's in genetic counseling and a doctorate in human genetics from Howard University, and she completed her postgraduate training in bioethics and the ethical, legal, and social implications of research at the National Human Genome Research Institute at the NIH, and in epidemiology and behavioral medicine at Howard University Cancer Center. Dr. Royal's research, scholarship, and teaching focus has been on the ethical, psychosocial, and societal issues in genetics and genomics, particularly the intersections with constructs of race, ethnicity, and ancestry. So let me welcome Ann Taylor to the podium.
Thank you very much, David, Charmaine, and to all of you who are here. I'm sure this is late in the afternoon for this conference, and that's the witching hour for conferences. So this session is, I think, of particular interest and importance to me. And when we start talking about race in medicine and race in clinical trials, I do think that, and I think you'll hear from Charmaine, the context and the use is the critical variable. Race and ethnicity is a little bit like water. Too much and it's destructive and we drown. Too little and we fail to learn important things. So just by way of introducing the substance of my talk, I'm going to talk a little bit about where we are in terms of demographics in this country. If you look at the population by race and ethnicity comparing 2000 to projected in 2025, we see dramatic changes in the demographics of the country. Leading causes of death by race and ethnicity are actually very interesting. They all cluster around similar causes. So predominant cause of death for most groups is cardiovascular disease, followed by cancers. A smaller group of clustering by stroke, but even within those clusters, there is variability by race and ethnic groups. Similarly, if we look at the trends in life expectancy by race and ethnicity between 1991 and 2006, the good news is that life expectancy is increasing for almost all groups. The bad news is, as we look at these groups, and these are groups who define themselves by self-identification, we see huge differences in life expectancy, and prediction of quality of life is the same. So when we talk about clinical trial populations, we really talk about variability, genus and differentia. And clinical trial populations vary in huge numbers of ways by sex, age, race, or the ancient continent of origin descriptor, ethnicity, which really defines common culture, language, religion, belief, many things subsumed under ethnicity. Etiology of disease also varies tremendously by populations. Comorbidities, although comorbidities are not necessarily independent of other causes of disease. Genetic background, and there's both the population genetic background as well as, perhaps more germanely and most importantly, the family genetic background. Epigenetics, which is really the study of how environmental influences or how environmental factors influence genetic expression. So one can see, for example, identical twins, exactly identical genetically, one of whom has congenital heart disease and the other does not. And the etiology of that difference has to do with subtle changes in perhaps blood flow, nutrients to twins during fetal life at critical developmental stages. Responses to health stressors, life circumstances, whether those circumstances are chosen or forced, care access, quality, beliefs, patient and physician access, quality, and belief and interactions. Socioeconomic advantage and disadvantage, lifestyle, cultural and behavioral habits, psychosocial constructs, both one's personal psychosocial construct as well as societal psychosocial constructs. And then something called health legacy, which is poorly defined, but examples have to do with, or one specific example is despite attaining certain middle-class status, we still see a higher proportion of infants, low birth weight infants in African American women compared to equivalently socioeconomically advantaged non-African American women. So all of these things can contribute to health, to disease, to outcomes. Some of them are independent of the so-called construct that we call race, ancient continent of origin. Some are very directly related to that, and some are mixed. So in one society, they may be quite relevant. In another society, they're less relevant. But all of these things have an impact on disease development, disease expression, response to treatment, and ultimately health outcome. So just to sort of rehash a little bit what we mean, or what is frequently meant by race or geographic descent. And that's really talking about the clustering of some common genetic characteristics which are associated with migration patterns. So humans migrate, they isolate, they migrate, they overlap. And this has been given strength by these genome-wide association studies which predict ancient continent of origin. However, this cannot and does not explain by a long way all of the differences in health observed when populations are categorized by this parameter and when these populations are compared. So let's just take a look at some variations in, and these are all taken mostly from US populations, differences in response to treatment, in disease expression, and in relative risk. So if you look at hepatitis C in response to interferon treatment, the response rates for patients initially infected with hep C were significantly different when African Americans have been compared to white Americans. Negativity in terms of viral load at the end of treatment occurred only in about 10% of African Americans versus 42% of non-African Americans. There are also differences in viral strain prevalence that differed significantly irrespective of the mode of transmission. And African American patients tended to have significantly higher prevalences of interferon-resistant viral genotypes. Lung cancer survival in the gefitinib trial and subsequent trial that use tyrosine kinase inhibitors. And here, the populations that benefited most were those of Asian origin, or if you were a never smoker. Now, this first trial, the ARESA trial, this analysis was done in a post hoc fashion, but the second trial of this class had a pre-specified subgroup of Asians, and again, the same pattern was observed. This is a drug that works far better in Asians compared to other groups that were in the cohort. And this was done with 210 centers in 28 countries. Interestingly, there were none from Africa or no subjects of African origin. Breast cancer is another place where we see differences in both incidence and mortality. So African American women have a lower prevalence of the disease, a lower incidence, but they have the highest mortality of all women. And so some of the increases in prevalence that are seen in other groups are related to the prevalence of the BRCA gene. But even that is modulated by age of menarche, age of menopause, parity, whether or not women breastfeed. The lesser prevalence of breast cancer in African American women, but a higher mortality. So some of that has to do with access to care, but there's also a higher prevalence of the more deadly triple negative phenotype of breast cancer. And we don't know what's responsible for this higher prevalence of this particular tumor type. But if we address, in looking at and deciding how we need to address this, we have to understand all of these variables in order to make an impact. Heart disease, the commonest cause of death in all populations, and you can see the distribution by both men and women. Black men and women are much more likely to die from heart disease than all other racial and ethnic groups. The risk of kidney failure is also not uniform by race. And if you look at the relative risk compared to whites, African Americans have the highest risk. Native Americans have also a very elevated risk, and Asians have a somewhat lower risk, but are still at higher risk of progression of kidney failure than are non-minority Americans. So one of the things that's interesting in terms of sorting out what might be the culprit in this, there is one gene, APOL-1, that's found in about 15% of blacks, but in 25% of blacks with kidney disease, and is almost non-existent in non-black populations. And if you look at the combined data from both the African American study of kidney diseases and the chronic renal insufficiency cohort, this gene appears to explain nearly all of the increased rate of the progression of kidney disease in black compared to white patients. So this is an example where understanding some of the genomic data is extremely helpful. What we will do with this is another story, but it is helpful to, in part, explain why this is really dialysis centers are populated almost exclusively, no matter what the community is, with African American subjects compared to non-African Americans. Similarly, if we look at responses to therapy, so this is data from the All Hat trial, which was a huge 45,000 patient trial with a very large component, about 15,000 African American subjects, 8,000 Hispanic subjects, and looked at endpoints of heart attack and death, all coronary heart disease, cardiovascular disease, stroke, kidney failure, and heart failure, comparing treatment with lisinopril versus chlorthalidone. Strikingly, if you look on the right-hand side of the panel, what you see is that in non-black populations, but the two drugs were not overwhelmingly different from one another. When you look in the African American population, there were very significant differences in endpoints using with chlorthalidone compared to the ACE inhibitor. The African American trial, African American heart failure trial, which was based on the old vasodilator and heart failure trials. In this trial, which was a very small trial, 600 patients with fortuitously, was published in 1986, with fortuitously about 28% African American subjects. When this was looked at retrospectively, there was a difference observed in the response to vasodilators versus placebo in black patients versus non-black patients. In VHEF2, which compared an ACE inhibitor to vasodilator without a control arm in this trial, what was seen was that the ACE inhibitor and the combination of isosorbide dinitrate hydralazine really were not measurably different in black patients, the outcome. And all of the advantage seen with ACE inhibitors was seen in the white patients in this particular cohort. So this led to the African American heart failure trial, which tested the combination of isosorbide dinitrate and hydralazine in a contemporary cohort of patients, all African Americans with heart failure, and all of whom were treated with ACE inhibitors, beta blockers, diuretics, digoxin, and about 40% of them received treatment with aldosterone. And what you see is the confirmation of the added benefit of this combination in a contemporary, well-treated heart failure trial, suggesting that at least in this group there is an added benefit of adding this therapy. So what are the consequences of the failure to include subgroups in clinical trials? And this has certainly been the common pattern until the last 20 years. Women, minorities, individuals of different ethnic groups were virtually not studied. So the lessened efficacy of beta blockers and ACE inhibitors in black hypertensives was really not appreciated until 10 years after the introduction of these drugs. Clinical endpoint data at the current time on ACE inhibitors in renal disease is absent in blacks until about a decade ago with the publication of the ASK trial, the African American Study of Kidney Disease. Data is still missing or uncertain for both beta blockers and ACE inhibitor benefit in blacks with heart failure. The tamoxifen breast cancer prevention trial had only 229 African American women out of 13,000 subjects. And this is in the context of recognizing that mortality is higher in this group, despite the fact that prevalence is lower. The guidelines that were based on those data from this trial recommended limiting the use of tamoxifen in blacks, where in fact no recommendation should have been done at all or made from this study regarding the use and the value of this therapy in African Americans. Some other examples of sort of what happens when you exclude subgroups, non-steroidal anti-inflammatories were mostly tested in younger individuals, but then used by the elderly where renal complications are high and really exacerbated by NSAIDs. Differences in coronary artery disease, risk, expression, and outcome in women certainly has not been recognized until the last decades with the Women's Health Initiative and the Women's Ischemic Disease Studies. So if we don't include subgroups, irrespective of how you define them, we lose information both on pathophysiology of disease and optimizing treatment based on the best evidence, which are randomized clinical trials. So how should we use race and ethnicity data in clinical trials? And so these are my thoughts, that population differences in disease prevalence, expression, and outcomes can provide clues to pathophysiologic mechanisms as well as clues to targets for treatment. Race by itself subsumes many, many factors. They're interrelated in complex ways, and in fact, some are sort of fellow travelers versus others being related to some common clustering of genes, to family histories. All of these things are interrelated and can't clearly be separated out, but race remains in this country a marker for outcomes, disease patterns, and sometimes for response to therapy. So we can't really ignore the health impact or fail to understand the reasons for these observed differences. So if we're going to optimize treatment across population groups and eliminate disparities, number one, we have to continue to collect high quality and large quantity data on health risks, disease expression, and outcomes in population subgroups. We need to use lots of different tools besides just randomized clinical trials, although that is the standard, the gold standard for treatment application. In fact, identifying differences is often done better through use of registries, administrative databases, observational studies, provided those observational cohorts are indeed ethnically and racially diverse. Large clinical trials are essential to identify differences in disease patterns. And we also have to use the evaluation of disease burden in subsegments of the population so that those segments are included in clinical trials, irrespective of how they're designated. So when we use these large databases, we need to also see where the disease burden is graded, and those are the subjects that need to be included in clinical trials. So randomized clinical trials with appropriate subgroups for statistical assessment of differences or similarities across these groups. We also need multidisciplinary research with appropriate subgroups and appropriate variables for statistical assessment of differences or similarities across groups. So if we only look at, for example, outcome of disease and we don't look at background variables, socioeconomics, where patients are cared for, patient beliefs, we're certainly not going to understand optimally how best to treat these patients. Where populations are observed, focus trials, which explore multiple biologic, social, and environment variables, can provide optimization for evidence-based interventions. So I think I'll stop there. And David, do you want to have questions for this portion or save this?
I think we'll
Perfect.
have questions at the end.
Okay.
Good evening, everyone. Good to be with you this evening. And I think you laid the foundation for me very well in terms of some of the things I'm going to say. There's a lot of overlap, but I have some other information to talk about. So I think our talks will complement each other very well. So the title of my topic is Race in Science and Medicine. And I'm going to talk a bit about how we understand race, what geneticists say race is or is not, and presented a lot of data about differences in populations based on racial categories. And I'm going to talk a bit about what that means, or question what some of that means in terms of what science and genetics tell us about race. So the fact that we're having this discussion about differences in outcomes based on these categories we call race goes back to a question that people have been asking for a long time. Does race exist? What is race? Are there human races? And geneticists have some answers to that, geneticists and evolutionary biologists. And I'm going to just, this slide just shows a bit of the media reports that have been out there over the years, and it continues in terms of biological basis for race and scientists saying there is no biological basis for race, and questioning why are we structuring populations based on race and what is race. And I'm going to show just a little bit about why there is the question about humans and race, and are there human races, and if race is biological or not. So genetic and fossil evidence have shown us that human populations originated in Africa, somewhere in East Africa, about 150,000 years ago, and moved out of East Africa over time, about 50,000 years ago, moved out of East Africa to populate the rest of the world. So what we do know about human populations is that there's a common origin, there's a common ancestry, a common place of origin, which is Africa. So human populations, from the evidence that science, genetics, and fossil and archaeological evidence have shown us, that we have a common ancestry in Africa. And because of that common ancestry, we see certain patterns in terms of genetic variation. So this is a slide by Sarah Tishkoff and colleagues at University of Pennsylvania. And it's showing variation in CD4, haplotype CD4 involved in immune response. And this slide actually shows variance in the three big continents that we tend to talk about a lot in terms of science and genetics: Europe, Africa, and Asia. And what we see in terms of those numbers represent variance, genetic variance in the populations. And we see that Africa has more variation than the other populations, but we also see that the other populations almost fit in Africa in terms of their genetic variation. And this slide is just one of many slides and many representations of data that we see when we look at human populations and genetic variation in populations. We see that Africa as a continent harbors the greatest amount of genetic variation, and that the variation that we see in most other populations are a subset of what we find in Africa. There are some geneticists and physical anthropologists that say the picture is even more dramatic than what we see here. Some of them talk about a nested relationship in which pretty much all the variation that you see in other populations, you can find it in Africa. So again, demonstrating the common origin, that we're more alike than we are different in terms of genetic biological variation. This slide is by Alan Templeton, a geneticist from 2002. And it really shows humans, I don't even know where to, I guess I can't, the pointer is not gonna, yeah, can't do my pointer. But what you see there, you see a bar graph with different organisms, and you see, well, this on the left-hand side, what you see there is FST, and FST is called the Fixation Index, and it's an index, it's an algorithm developed by a geneticist, Sewell Wright, in the early 1900s. And it is an algorithm developed to look at population, to measure population variation, population differentiation. And what we're looking at in the slide on the x-axis is different organisms and their fixation index. And the one that's highlighted there is humans. And a fixation index of 1 means that the higher the fixation index means that it's more likely that these populations can have separate populations within them. So race, in terms of, let me just define, in terms of what evolutionary biologists and many human geneticists define race to be, is subspecies. So race is a level of differentiation below the level of the species. So you have a species, And then you have distinct evolutionary lineages to form different species or subspecies below that level. Those subspecies are what are called races in terms of evolutionary biology. When we look at these organisms, you see gray wolves on the end there and white-tailed deer, and they have a high fixation index. And those organisms are likely to substructure into subspecies. And humans, so the threshold that Sewall Wright set is 0.25. 0.25. So organisms with a fixation index above 0.25 are more likely to have subspecies or biological races. Organisms with a fixation index below 0.25 are less likely. You see where humans fall there. They're pretty much lower than most of those organisms because the fixation index, when we look at human populations, we don't see distinct evolutionary lineages. We see common ancestry, and we see populations that are more alike, and they pretty much stay alike. They don't separate. They have not separated over time into distinct subspecies, or what we call biological races. There are people who ask the question, so, because humans as a species is much younger than many of those other species, or relatively younger. And so there has not been enough time for population variation to occur. In addition, humans don't remain isolated. We move, we mate. And so variation, it does not get, the separation does not have time to occur because people move around. We're not isolated for the most part. So there are four things that geneticists talk about and evolutionary biologists identify, and there may be others, but these are four key things, four key pieces of evidence that tell us that humans do not subdivide into biological race. And I keep saying biological race because that's the terminology in terms of biological race, because I will talk a bit, and Anne has talked a bit about what we call social race. And we'll get into that discussion, I'm sure, when we have our conversation. So there are these four things: geographical location. So when we look at geography, we look at human populations in different geographic regions, we see really a continuous variation across populations. We don't see discrete units, meaning Africa being separate from Asia, being separate from Europe in terms of genetic variation. We see continuous variation in geography when we look at human species. When we look at gene frequency, frequency of genetic variation, we see more genetic variation within any of these populations that we call races. There's more genetic variation within them than there is between them. About 85% of the variation in terms of genetics that we will find, we can find it in any one population. There's only a little bit of difference from one population to the next. Morphology, one of the things that we use a lot to point out races and to identify races, how we look, our phenotype. And when we look at skin color and eye color and hair texture and all these things that are markers of race, we don't see them traveling together genetically necessarily. They're not obligatorily linked to one another. And even more importantly, they're not obligatorily linked to diseases or other health conditions. So we have what we tend to call non-concordance of human traits, in that we don't see them always staying together. We see people who are identified as black with blue eyes. We see people who have straight noses who are identified as you know, with one group that we wouldn't associate straight noses with. So we see variation in terms of these phenotypes. And then lineage, as I talked before, we don't see distinct evolutionary lineages in human populations. It's one lineage, common ancestry, and we're all members of the same family. So then what is race when we ask the population? So I just talked to you about what geneticists and scientists say race is. This is a word cloud from a study that I'm doing now with a national survey that we did of the US population. We're still, this hot off the press data here. We're still analyzing these data. My postdoc did up this word cloud, and I thought I'd just show this. So this is what we got when we asked people to define race, right? They're all over the place. And for those who, I'm sure everybody here is familiar with the word cloud, the words that are larger are the ones that were most common. So most people, when we ask them what race is, most people say color. And that's what we find a lot in studies that ask people about race. The first thing you hear is color. Many of the studies that have been done with the general population asking people about race, we hear color. What we found in this study also is that there seems to be more confusion, not so much confusion, more varied response when we ask about ethnicity. People answer more similarly when we ask about race, right? And they use ancestry, they use origin, they use these terms. When we ask about ethnicity, people are all over the place. Some people actually say, I don't know what my ethnicity is. Ethnicity for some people seem to be a very, very, very complicated term. So in terms of the US Census, and Anne talked a lot about the OMB categories and how they're used in research and the trends that we see in terms of health. The OMB categories are required by the NIH and the FDA for us to use in studies. And we have the 1993 Revitalization Act that requires us to include women and minorities in studies. And we have the 2001 NIH policy for reporting based on these categories. So these categories in the US have become quite important. But the questions about what these categories even mean, and these are the race categories, and you know we have two categories that we call ethnicity categories, the Hispanic and non-Hispanic. But the census is actually in the process of modifying, exploring changes to the census, to the 2020 census, really based on the numbers of people that are checking some other race. They don't identify with any of these, and Hispanics and Middle Easterners and North Africans fall largely in those categories. So they're looking now at combining the race and ethnicity categories. So rather than having race, what is your race, and then you saying I'm Hispanic or not, Hispanic ethnicity is gonna, or Hispanic is gonna be one of those. So it's gonna be 6 of those. They're exploring that. We'll see if that's what's gonna happen with that. I could go on a bit about the census, but I'm gonna move on because I wanna get through. So when we talk about racial medicine based on race, there are some assumptions, and many researchers who use race in research don't intuitively think about these assumptions. But when you read studies that have looked at racial groups and look at differences in groups, there is something about many of those studies that assume that there are biological human races. When we look at differences, how the studies talk about their results, how they publish their findings, a lot of the times lead you to believe that these groups really have some biological difference. And I'm not saying there's no difference, we're just saying that they're not racial differences. They don't they don't rise to the level of race, in that the variation that we see is so great. And that races exist, that they differ in their genetic composition, and that these genetic factors are what lead to the differences in health. And that's the slippery slope when we start looking at differences and then not careful about including a lot of the things that Anne talked about in terms of other factors as opposed to just looking at genetics. Again, other factors included, this is a model I developed with colleagues when I was at Howard. Very complex, everything plus the kitchen sink is in here, looking at different factors that influence health outcomes, based on a lot of data on African Americans, but really can be extrapolated to other groups as well. So that looked at factors that influence health and disease. And this I got from Merck in terms of factors that influence variation in drug response. There are lots of factors. We know genes, we know genetic variation influences how people respond to drugs, but there's a range of factors that also influence that. So we need to start designing studies to be really more integrative. And Anne talked a little bit about BIDEL, and this is a picture that I took earlier this year at the American College of Cardiology meeting, where I conducted a small survey of cardiologists on BIDEL. I'm going to show just a little bit from that. But this is a poster by Arbor Pharmaceuticals, who now owns BIDEL. I know many of you who've heard about BIDEL, you knew that Nitromed, the company, went out of business. But BIDEL is not dead. It's very much alive. and this company, Arbor Pharmaceuticals, is marketing it. And I was chatting with the guys at the booth and took the picture, and lots of interesting things going on there. But in a study that I did with my team last year, I think, with cardiologists in the Association of Black Cardiologists, one of the organizations that was involved in the AHIP trial, a small study that we did. These are some of the things that we found when we asked how people were using BIDIL. I think BIDIL is a great example of how we think about race in medicine. BIDIL did not include genetic data. The study itself did not include genetic data, but there are a lot of biological and genetic implications there just in terms of how the study was done and the conclusions that have been drawn. So these are some of the things that we got. Many are using BIDIL. Cost was, at that time, cost is a major issue. Race was not the primary thing that many of them used to determine who gets BIDIL. They use symptoms, indications, which I thought was really interesting. They don't only give it to black patients. Which, chatting with them, some of them really said, you know, I give it to everybody, which has been one of the arguments about BIDIL, and we can talk about that later on. And then these are a couple other things. Earlier this year, that we also did a study of cardiologists at the American College of Cardiology meeting on BIDIL. And this is a quote from one cardiologist. I think it is too simplistic to do race-based therapy when asked what he thought about BIDIL. Because I think right now there's more personalized medicine, and that we could do a whole talk on personalized medicine and what that even means, genomics, et cetera. And I think that is probably a smarter way of going about it. So he talked about genomics, but we all know we can't just look at genomics. As much as I love genomics, fascinating science, it doesn't have all the answers, and we need to have more holistic approaches. This is a different quote from a cardiologist about something that he hears a lot from his white patients. Give me that black drug, it works for me. Which again, in his mind, is like, I give it to everybody. Everybody needs to take this drug. And then there are some black patients. I remember some years ago, I interviewed an African-American man who has heart failure, who refused to take BIDIL. And when I asked him, why won't you take BIDIL? He said, they made that just for us. There's got to be something wrong with that drug. I'm not taking it. So he refused. His doctors recommended it, and he just would not take it. So I think when we think about race and health, and BIDIL, for me, brings this to the fore. Scientific validity. What does science tell us about race? What is it? What is it not? And social justice, right? So we know that racial categories and race in America was constructed, and we know it was constructed for all kinds of reasons. And because of that social construction, we see different health outcomes, right? It's race, for the most part, that has resulted, the construct of race and what has happened to people who have been racialized that results in what we see. But how do we address those issues, still bearing in mind that these groups are not biologically homogeneous? That is a challenge. That is a challenge. So I'm going to wrap up my 5 minutes, David? Yes. All right. By talking a little bit, and I hope we get time to discuss a lot more. I'm just going to talk about two things in terms of what all of this could mean for IRBs. I know many of you are members of IRBs and use IRBs for your research. And I'm going to talk about the issue of inclusion and methodology, study design. So inclusion, that's one of the things that Anne talked a lot about, the fact that many studies do not include diverse populations. And as IRB members, you get protocols from time to time where people just don't have enough diversity for the question that they're going to ask, and I'll get to the question in a minute, but there's not enough diversity. And you need to recommend, or should, that they do whatever they need to do to get more diverse samples. But even though I agree and I know that we need diverse samples so that our results can be more generalizable, not that the results that we get from samples that are not diverse will not be applicable to those people who are not in those studies. That's not the point. It's that our results would be more generalizable to a greater number of people. But I also recognize that every study does not have to have multiple groups. We don't always have to have a comparison group. It is perfectly fine, and I think we need more of those studies. It is perfectly fine to study one population, one quote-unquote racial group, because I don't think we have real racial groups, because there is variation within those groups. And I think the more we look within the groups is the more we will dispel this notion that we always have to be looking between the groups and comparing. There are African American men who get prostate cancer and African American men who don't get prostate cancer. What is going on there? We don't always have to look at black men and white men to find out what's going on. We can look within the group because there's a lot of diversity. There are a lot of differences in behavior. There's also genetic variation within the group. So I think we need to do more within group studies than we are currently doing. And the issue of inclusion, and it would just take me to my next point, depends on what the question is. It depends on what our question is. So the question needs to drive the study design. So you're not just going to look at a study and say, oops, not diverse, they need to go back to the drawing board. What is the question? What are they trying to find out? And then in terms of the question, that determines what kind of study design, what factors do we look at? Do we need to look at genetic factors? Do we need to look more at social factors? Do we need to have a more integrative model? It's fine just to look at a genetic factor that might predispose people in a population to a particular outcome, but how we write that project and how we talk about it and how we publish it when we find that this genetic variant is more common in this group than another group, what does that even mean? So our study design and how we report our findings, I think, becomes part of the IRB's job, because we really need to make sure that the studies don't perpetuate these notions that there are biological races. I'm going to stop there and looking forward to discussion.
So thank you very much, Charmaine and Anne. And we'll take questions now. So if you have a question, please step up to a microphone. And don't be shy. Sir.
Yeah, I'll start. You hear me okay? Oh, there I am. Okay. So I don't sit on an IRB, but I do participate in a number of community advisory board entities. And could you, either both or one of you, comment on the influence of those types of boards and how they can help with either protocol design or with expanding this notion of inclusion?
So I think Charmaine and I said a lot of the same things. She said them more elegantly than I did. But I think the focus in constructing trials is where is the burden of disease? And are those populations with the burden of disease included in the studies? Traditionally, we've seen this with women. We've seen it with other ethnic groups where there is tremendous burden of disease, but they are not included in the trial. We do not learn about either the genus and differentia of pathophysiology, or we don't develop an evidence base to optimize treatment.
Yeah, I think community groups, community advisory boards could also be very helpful in helping us even frame the questions. I think for a long time, and still, researchers come up with the questions and go into communities and go into groups and do our research. And that's okay, that's what we do as academics. But sometimes it would benefit from some input from those communities in terms of what's important to them. And we are not always going to be able to do what's important to them, but there has to be dialogue. And in terms of the research, but also how we conduct the research and how we report the findings. Community groups can be very helpful in working with researchers to do those things.
Thank you both. Jay Smart of Miami University. I'm going to ask questions sort of as I'm chair of my IRB, so it's kind of an interesting place. The thing that struck me, is, Dr. Rose, about the social justice aspect, because one of the things I worry about is that it's kind of inclusion too, I'm trying to pull this all together. It's the idea of people wanting to do research, particularly around race. And being careful about not letting, say, personal issues cloud sort of the science issues. So, for instance, we have a lot of researchers, a lot of our social group is interested in looking at issues of race and prejudice, and that's kind of their group as a group. But we, one, don't really have the population to do that kind of research well, I think. I think it's the same idea where you have to sort of seek that out. And two, I think there's kind of a blind spot for a lot of research in terms when they propose it, scientifically, it's fine. Like it's the right design in terms of methodology. Yeah, one group gets this, one group gets something else. But not realizing that there's an implication of that for your participants. So to give a quick example, one of the ones that we spent a lot of time talking about was wanting to look at perceptions of humanness or something like that. They're basically trying to see if there was an actual perceptual thing that tips people off about race or in-group, out-group. And what they wanted to do was morph person faces with faces of chimpanzees.
Yeah, I know those.
And for me, that raised hackles just right off the bat. And I was trying to divorce because that's a bad association to have. And it took us a good 6-7 months to sort of work out a plan for that. But I understood where they're, like, these people weren't, like, they're not like an evil doctor or something like that. It was just it was their interest was looking at, you know, is this really in-group, out-group, or is there something else that's causing this? But it was also a blind spot in terms they just didn't recognize that this was going to be problematic, particularly for participants for that. So I guess my long story for probably a short question is how to navigate that aspect of it, where some of the research needs to happen. So I think the dialogue has to happen, particularly with recent events. But balancing that out with, you know, it's interesting for somebody, but it's somebody else's life. And so how do you balance those kind of two aspects of it? Because I think it does need to, we do need to talk about it. But how do we, particularly as the IRB, it's like I don't want to just shut stuff down because it's uncomfortable. Right. But
got it. Thank you.
That's a tough one.
Yeah. Sorry.
Gosh, I don't know. Anne, do you have anything faster than me?
No, I was hoping you were going to take that one.
Gosh, I think.
I could comment if you need to.
Help me out, please.
You know, I think that is a real issue that IRBs struggle with in a kind of day-to-day way, and it's really in certain ways it's a small issue compared to the larger issues of identifying treatment responsive issues or disease-specific characteristics of race. And it's much more about thinking about the impact of the research or even the research question on the study subjects. And to identify sensitivities, I think, requires either the input of an appropriately sensitive IRB or the involvement of an appropriately constituted external advisory committee. And I could think of a lot of examples in which the questions that were asked, I mean, we normally, in a free society, don't think you can't ask a question. You should never, we like, as you're suggesting here, we like to be able to ask and answer questions in a free academic environment. But nonetheless, the way certain questions are asked do add stigma or burden to the populations. And I think we need to carefully assess whether there is enough scientific value in the question or in the study as designed to approve it at all, and then to think about whether there's less, and I'll use the word burdensome, less burdensome ways to get that question answered. And I could think of some examples that we've struggled with, but we have other people asking questions, so we can move on. Do you want to respond to what I said there? Or?
No, I agree. I mean, I think bringing in an external group or person or couple of people to help you think through what the potential issues are, people from those communities who might be affected by the research, I think that's a way to go.
Yeah, it's difficult to, you have to be careful not to ask questions inadvertently too, like people who study criminal behavior or violence or other things that have been too often historically associated with vulnerable communities. And so You know, that's the other sensitivity that needs to be brought to bear on some of these questions. Anne?
Yeah, I was going to agree with you about that. One of the hugest problems is sort of juxtaposing or joining qualities, characteristics that have no real relationship to one another. And we attribute those things as being causally related when they're completely unrelated. We need to be able to explore population differences, but we need to be able to do it in a way in which we're looking at differences, not who's superior, inferior, or making the assumption that difference in arena A automatically means difference in arena B. And that's the, I think it's a problem of this society.
Hi,
Sure.
Mark Robson from New York. So I do a lot of genetics work, and I wholeheartedly agree with what you were saying about there being a lack of genetic basis for social race. But there clearly are genetic subpopulations that are defined through ancestry informative markers and things like that. But putting that aside, it seems to me that this is a question of eligibility for whatever the research question happens to be. It's a definition of population. And do you think that there are circumstances where socially defined race is an appropriate eligibility criterion for a study?
I think there is. And to go back to your point about population differences, yes, I certainly agree with you that we do see variation in populations based on ancestry. But I guess the point that we've always in genetics made is that that does not mean there are races, because races means a level of homogeneity that we don't see, right?
Imperfectly
But
correlated.
I think when we talk about social race, or the groups that the U.S. calls race, yeah, I mean, we see differences in outcomes, and we still need to study why we have those differences. I think a lot of the study designs, though, are problematic to help us understand those things. So I think these groups can be involved in studies to study different things that we want to study in terms of differences that we may see, or again, to look within these groups to see what's going on. But I think, for me, at the heart of any of those studies is the study design, is the question, is how we do study those groups and what we're looking at.
So if I might follow on, how would you advise an investigator to make those groups, and to define those groups, who wanted to look at differences?
All of you are asking me these hard questions.
I'm actually genuinely want to know.
I'm just kidding.
I'll tell you a story afterwards about why.
The issue of how to study populations is the big challenge. It's always been the challenge. People talk about self-identification, and that has its place, because people who define themselves in similar ways may have similar experiences or not. I think so there are times that we need to look at those groups. I think a lot of times when we are looking at what we call racial groups, we probably need to be looking at ancestral groups, or people with more similar ancestry than people who have more similar social experiences. So I think it just depends on what we're trying to find and how we look at the group. So for example, the group that we call Black or African, the group that's called Black in the US, includes people from Africa, people from the Caribbean like myself, and we're all lumped together as Black people, but have different experiences, have had different experiences before coming to America. Maybe once we get here, they become a little more similar, but have had some different experiences. So I think there's a place to tease those out, depending on what we're trying to find out. If we're trying to find out about cultural beliefs and practices, then we may want to tease those apart. If we're looking at other things, then there may be a place to lump us all together. So I think the question for me still drives what we do, whether we disaggregate the data or whether we lump them together.
You know, I agree with that completely. and I can't believe a cardiologist who's basically a surgeon that doesn't like getting up early in the morning is going to talk about sort of social issues with someone who's much more of an expert. But I still think that we need to look where disease is. We need to have large databases, and how people, if the majority or large numbers of people with a certain disease fall into a certain group, we have to focus on that, regardless of what we call that group. We can look at ancestry sometimes, but ancestry is not uncoupled with all of the other events that happen in a person's life. So it's become a little bit fashionable to say continent of ancient origin, but if your continent of ancient origin is Africa, your life experience in Europe and this country is very different from a person whose continent of ancient origin is Europe. So these things often can't be uncoupled, but where is the disease? Who has the disease? What distinguishes the characteristics of that disease? And then, again, the question is, what do we do about it? So the question does drive the selection of the population.
Yes.
Good afternoon. I'm Fabian Sandoval. I'm with Emerson Clinical Research Institute. We are 7 sites within the DC, Virginia area. I have access to approximately 180,000 minority patients, Hispanic and African Americans. As we conduct research, I get this question, and I have a canned answer for this question, but I'd like to hear what you would say to this question, which I've heard from new and existing investigators and from some IRBs. When we are looking to conduct studies, they say, well, Hispanics, because they are migrant workers and some African American cultures as well, they're considered a vulnerable population. So let's think about that when we're doing this study, because they're a vulnerable population. I have an answer to that, but I really would like to learn from you on the panel to see what you have to say to that question. Are they a minority population? I mean, vulnerable population.
Vulnerable?
Well, so Hispanics aren't a single group by any means. And if you look at population variability and health variability between Mexican Americans and the East Coast, Hispanic from Caribbean, from Central America, from Argentina, very, very different profiles. So again, lumping somebody by language, it's like looking at everybody who spoke English. That's a hugely diverse group. So what you're really interested in, I think, is, and being a vulnerable population is less by virtue of language status than by social factors. And so those are two things that don't necessarily need to be coupled.
Right. Yeah, because I was going to say, it depends on what you mean by vulnerable.
So they say, well, they're uneducated. They don't know how to read. They're vulnerable.
Oh, that's kind of
They don't have,
lumping
I'm
a
sorry,
whole
I
group
missed.
together. He said, saying they don't know how to read. Boy, that's not stereotyping. I don't know.
That's the generalization that I get from some IRBs. I'll get that generalization, or I'll get that generalization from research committees that are interested in doing the research, but they say, oh, well, they're a vulnerable population. So my answer to that,
but they're a vulnerable population, so we can't study them, or we just have to be careful?
So we have to be more careful with conducting research with them, with us, because I'm a minority as well.
So
That's sort of a strange construct. And, again, I think you look at racial origin or national origin doesn't necessarily couple with being vulnerable. Vulnerable has to do with not being able to understand and comprehend.
I'm trying to get some validation, because I'm on my own kind of trying to fight this battle in the metro area sometimes, but when I come to a conference like this where there's experts in the room and on the panel that kind of agree with me, it kind of, I think, is giving me a little bit more justification to what I say, which is my answer is, based on, CFRs, right? If you look at subpart B, C, or D, those are the vulnerable populations. I don't see any vulnerabilities in my patients just because they may not be able to read or understand, or their ethnic background for them to be considered vulnerable. I think the question has to do with, is there an issue of coercion are these patients being coerced into being a study? That's a better question for,
Yeah.
for them.
I mean, and this is a question, I guess, for everyone. Are minorities in general considered vulnerable?
No, no.
Okay.
I mean,
All right.
vulnerability has to do with individual competency to make a decision to participate. It doesn't have any ethnic, racial, social boundaries. It's individual competency to understand and participate.
And I've heard that from IRBs coming to me. I've sat in front of IRBs, and IRBs have said to me, 2 IRBs in particular had said to me, well, they're a vulnerable population.
Well, there are times that people use that term vulnerable, which is why I asked the question. I think there are many people who just include minorities, whatever that is, in that vulnerable population category.
That's right. I think it's a problematic concept, and I think we talk about vulnerability, as you were saying, as vulnerable to coercion or undue influence. And under the regulations, it means you have to have additional safeguards in place. But by equating minority status in the DC metro area with vulnerability in terms of needing additional protections, I think is casting not just a too wide but a problematic net. On the other hand, what I would say is if you know your population is undereducated and immigrant, for example, and they may have certain concerns about interfacing with the medical establishment, that the protocol needs to be tailored to their needs. But certainly, there's no cause to exclude them from research. I mean, you know, Ann and I was going to say live and work. It sometimes feels that way. We work in Washington Heights, where the population is Dominican and often largely first generation or new immigrants. And it would be incorrect and foolish not to understand that when you're designing your consent documents and even designing your studies. But the notion that somehow we would, in a blanket way, exclude them from participation or say that they were too vulnerable to participate is not something that we would consider, I think.
Thank you.
Let me take the next question.
Okay, I have a comment because I think you misspoke. If a person can't understand a consent form or a research study, then they are vulnerable. But if they can't read, or if they were a person of color, that's not necessarily a vulnerability. I mean, not being able to read makes one a little more vulnerable, but there are ways to compensate for that through the consent process. But I just wanted to make the difference between not being able to understand, because we're struggling with, at my institution, participants with early stage Alzheimer's disease, and whether they can, they have the ability to consent is really fresh in my brain. But those patients we may consider vulnerable, but not someone who can't read per se. But my real question is, I struggle with my IRB making arguments to include underrepresented groups because of the potential to find differences in treatment outcomes or differences based in genetics that may have origin in geographical origin, which is a phrase, Dr. Taylor, that I like and I'm going to take back to my institution because they don't seem to see the value in being more inclusive. And we study, I'm from New York City, I'm down the road from you all, and we study some diseases like some kidney diseases, HIV, and some other diseases where geographical origin and mutations that result from encounters with diseases based on geography in the past may affect treatment outcomes now. But I can't seem to get the need to be more inclusive across? I mean, people shake their heads and say yes.
So let me rephrase the question. So what you're asking, and I think it's a question that I think a lot of us struggle with, is an IRB obligated to ensure that the sample is broadly representative? In other words, that it includes subgroups and underrepresented groups in the population to permit specific analyses?
I'm asking, how do you make that obligation palatable and move forward?
Okay.
Yeah, no, I think that, again, what's the question that the study is trying to answer? What's the population that has the disease should be the determination. And that I really do feel strongly that we need to push and make sure that those populations that are burdened by the disease are actually included. You know, for years, many, many decades, all studies in cardiovascular disease were done in men of European descent. And we missed an enormous amount of pathophysiologic data as well as appropriate treatment data. And this concept can be extended to other populations of variables. There's something I want to talk to Charmaine about a little bit later, which is about the term racial and ethnic. Whatever the population, the variant of the population is, if the disease burden is significant, those groups need to be included in trials.
No, I agree.
Mm-hmm. So my question for Charmaine is, you know, I think one of the problems is we're not really saying different things. The problem is, how do we talk about population variability without using the word race? Because race is so fraught and laden with such destructive connotations and denotations. And even though we're 99.9% alike, that 0.1% that's different can be responsible for differences in pathophysiology, disease expression, and response to therapy. And I've often thought, having the experience with the African American heart failure trial and other studies, that if we could find a different word to describe population variability, that we would do ourselves a big favor.
And that's part of my mission.
So you know,
stay in touch about that.
it is 5:30, and I think it is time to stop. And I think that's a good note to end on. That what you were just saying. So I want to thank you all for your involvement today, and I'm sure that Anne and Charmaine will be up here if there are a couple of other questions. Thank you.